Prima parlerà la dottoressa Zobor, sempre dall'Università di Tubinga, siamo felici di averla qui, e poi avremo l'altro relatore da Londra.
So my name is Dott ssa Zobor and I'm also very happy to be here. I think I already know some of you, which makes me even more happy. And today, as a clinician, as an ophthalmologist, I would like to talk first about the disease itself, how we clinicians see it, and what the possibilities are when you are making a correct diagnosis. I will try also to explain the symptoms, why they occur, and I will also refresh the memory a bit, just like Susanna Cole and Christiane Marchesa did, by just taking a look at the anatomy of the eye and just giving you some information on the disease. But I'm pretty sure you are probably very aware of these numbers. So, as we've already heard a couple of times today, achromatopsia belongs to the orphan diseases or rare diseases. 1 to 30,000 is the prevalence, which can be calculated here. And the inheritance mode is autosomal recessive. This is what has already been discussed today in detail. The most important thing is in achromatopsia that it is a lack of cone photoreceptor function. I know that some of you in the previous years might have heard that cones are not really existing in the retina, but this is actually not true. It's only the lack of cone photoreceptor function which causes symptoms. But as Susanna Cole has already mentioned, during the years we do see a very slow degeneration and a loss of these cone photoreceptors. The achromatopsia disease can also be called as total color blindness or monochromatism. This is what we've already discussed today, and complete or incomplete forms occur. This picture is also pretty much informative because we have to deal with the eye, so we have to refresh the memory what biology has given us. When light proceeds into the eye, it falls at the back of the eye onto the retina, and the photoreceptors, the cones, and the rods will be the first site which just gets the information. They have to perceive this information so that it can be forwarded to our brain and that we have a vision. If we take a look at the eye, as a clinician, if I look into your eye, I see the retina at the very back of the eye. I also see the very center part of the retina, which is called macular area or yellow spot, and the very center is called the fovea. I also see the optic disc. This is where all the nerve fibers leave the eye and take the information to the brain. And of course we see the vessels, which are giving oxygen and nutrition to the retina. This has also been shown and discussed today that two different kinds of photoreceptors are distributed in the retina in a pretty different way. At the very center part of the retina, we have only cones. And this is the place where we have our most cones. If we go to the periphery in the retina, we have a smaller amount of these cones, but they are existing there also. The periphery of the retina mainly contains rods, which you can see on this curve. So if you take a look onto the retina, you can see that the very center contains only the cones, and here you have cones and rods, while the rods are even much more regarding the numbers. They do have two very different functions. The rods have to be used, they are used in very dim circumstances or even at night because they are very sensitive to light, but they can only see at one
wavelengths the best way. So there is only one type of these rods. In comparison to that, we have fewer cones, but we have 3 different kind of cones, which enables us to see colors. This is what Susanne Kohl has already told you. And these cones are also very important for us, that we have a daylight vision and that we have a very high resolution at the central part of the retina, where we have most of our cones. If these cones are not working, then we are talking about achromatopsia. The genetic background of achromatopsia has been discussed in detail, so I'm not going to get here into detail again. But as Susanne Kohl has already mentioned, CNGB3 is the most common form. CNGA3 accounts for 20 to 25% of all cases, and there are some other mutations where we only know a few cases. All of these genetic problems occur in the visual cycle in the cone photoreceptors, so if they are not working, we usually have similar symptoms which you as patients are aware of. These are what also describes the disease as total or incomplete color blindness, but I think most importantly the photophobia, the jittering eyes, so the nystagmus, and the reduced central visual acuity is what really disturbing, because this glare which the patients, which you have to live with every day, is very difficult under daylight conditions. In these cases where the light is very bright, your rods are really bleached, so to say, which enables a proper vision. They are not really used to such circumstances. Therefore, it is very difficult to orientate yourself, and there are some tricks you have to use that you do get some visual information. What we know about the disease, that this is a stationary disorder regarding the function, so it occurs at birth, and the function does not really seem to change. But we do know now that there are some morphological changes, some changes in the retina, which can be obtained during the years. If I want to visualize it for some of us, it's like an out of focus, overexposed black and white film, and this overexposed picture is the most important thing, I think, in achromatopsia. This was written by Caitlin Smith, who is a singer and songwriter, and she is also suffering from achromatopsia. She's from Canada. If we want to imagine how achromatopsia patients look like, we see this picture. We don't really see sharp images, so the visual acuity, the resolution is bad. Everything is black and white or different kind of shades of gray, and the total picture is overexposed, so we cannot really see the details. How do we characterize this as a clinician? There are several possibilities for that. The one group is a subjective measurement, so these are all measurements or examinations where we need your help. We are depending on how you respond in these tests. This is like measuring the central visual acuity, but this is only one parameter when we want to describe how patients see and what vision is in these cases. The other thing we can do is that we can measure test color vision. There are several kinds of tests, and I know that there are some tests which are quite confusing and really disturbing for you because you not really know how to solve the problems. But there are also special tests which we can do in achromatopsia, and we can get to a proper diagnosis, and I think this is also important if we are, as clinicians, looking at our patients. We can also measure contrast sensitivity, or we can also measure the visual field. However, in visual field, sometimes regarding the blindness, it's very difficult to perform really good tests. This is for you sometimes very difficult because we have a standardized background illumination, and even that background is pretty bright for you. Therefore, you have to squeeze your eyes, and it also results in visual field changes which are not really there, but you do are suffering from. What we can also measure is dark adaptation, and this is an information regarding the remaining rods which are still working properly in achromatopsia. However, we clinicians do need some tests which we can do to see objective results. So where it's not a test where you do something differently or you cannot properly do. These are tests like electrophysiology, which I'm not going to talk about in detail because Professor Andreasen is going to talk about that. And pupillography is also a measurement, which is also an objective test. However, this is not a routine examination. Besides the function, we are also very interested how the retina looks like and what we can see, because it is also very helpful when making a diagnosis. If we take a look at the eye of an achromatopsia patient, even if it's a young achromatopsia patient, we usually see that the anterior segment of the eye, so the cornea, the lens, and everything is looking fine. Even in most of the cases in young ages, we do not see any pathology at the back of the eye. So the retina, the optic nerve, the vessels all look pretty normal, but still we have a function loss which we observe in our functional tests. If we take a look at the eye of an adult patient or an elderly patient suffering from achromatopsia, we do see some small changes, but they only occur in this very central part of the retina. It's rather unfortunate because it's also the very center part of our retina, which has the highest resolution. Because the cones which are sitting here in the retina are not functioning, you might not even notice that there are some small morphological changes happening during the disease. Now, one appearance is when we take a look at the eye, but we might not be able to see very small changes. Therefore, we have to do further examinations like the optical coherence tomography, or as shortened, the OCT. This is kind of a test which is available in most of the clinics nowadays, and it has been developed in the 1990s, and it improved in a huge way in the last years. So we are really able to see the retina, the different kind of layers, in a very high resolution image. So we can also see whether the photoreceptors or the other cells in the retina look normal, or small changes can also be detected. This is also an examination which is non-invasive, which means you just take a picture from the back of the eye. This is a short procedure, and it can be done even in cases where we have problem with nystagmus, or where we have problem, for example, in children. This is also, I think, possible, although I have to say that the person who is doing the measurements also has to be an expert, because you need some patience regarding nystagmus to be able to do a proper image. If we take a look at some of the examples, what we see, you can see here the color pictures of the back of the eye, which does not really show us any pathology. These are special images from the retina called fundus autofluorescence images. They give you information about the retinal pigment epithelial layer. This is underneath the photoreceptors, and these cells work together in very close connection with the photoreceptors. So this is also very important as an examination regarding morphology. Some of the examples show here how the retina can look like in achromatopsia patients. For example, here on the right side, you can see that the retina looks almost normal. But in some, or even here, but still maybe some very distant changes occur in the very center part. While other patients can even show that there is a functional, not only a functional, but morphological change in the cone photoreceptors, so in the very center part of the retina, and this can be even bigger. There were a couple of studies which could describe changes in achromatopsia patients, what we could see in OCT images. And these are the typical changes we observe. We can observe the loss of the cones in the very center part of the retina. We can see a kind of an intraretinal bubble, which I've shown you in the previous picture. We can also see that underneath these missing cones, an atrophy, a degeneration of the retinal pigment epithelium can also occur. And in some cases, we do see a foveal hypoplasia, which means that the very center part of the retina is not built properly, so it's not developed properly. These changes occur in different percentage, and if we take a look at this study, we can see that very young children, or even the first 2-3 decades, or 4 decades, we only see changes and very small changes in the cone photoreceptors. In small children, we might see 1 out of 10, but as they get older, we do see a small further progressive degeneration in these cones. And as our patients get older, we can see that the retinal pigment epithelium is also disappearing. These are marked here as a black color. So what we can conclude is that there is a very slow morphological change of the central cones, which means that is a very slow but progressive disease. We have a loss of these non-functional cones. However, the function remains the same over time because it was absent from the very beginning. Now, OCT is only one kind of way where we can see in a very high resolution the cell layers, but it does not give us proper information how each cell could look like. For that, we have another possibility, although it's kind of a new method to see the retina and to see the photoreceptors, and it is developing also, has been developed also in the recent years, and the resolution is getting better and better over time. This is called adaptive optics, which is a technology used for astronomical telescopes or microscopes, and in the recent years it has been applied for retinal imaging systems also. This is a system which can correct optical aberrations by using a deformable mirror. And if we don't have these optical aberrations anymore, we can get a really high resolution. So you can imagine that we can see each of the photoreceptors actually. It is, though, very difficult to perform such images, but I do think that this is reasonable, and this is a way where we have to go if we want to get more information on the disease and on the morphology as possible. Now, there have been some studies regarding achromatopsia, how the photoreceptor mosaic looks like in adaptive optics measurements. And what we can conclude that this is, for example, a normal retina here in the very center part, and this is 10 degrees from the middle into the periphery. What we can see that the photoreceptor mosaic is changed in achromatopsia patients, and normal wave-guiding cones are not there. But we do see that there are cone structures which are still there, so there is a significant hope that these cells can one day be treated in gene therapy trials.
this is what I also like to talk about on this place, gene therapy trial, and just jump into this next huge part actually. There is a lot of information regarding genetics, what we already know, and ever since genetic diseases have been described, there was also the idea that we have to try to repair these DNA segments. And in 1998, Susanna Cole has described the CNGA3 as responsible for achromatopsia patients, and ever since in our labs and also worldwide there have been a lot of models, and also of course animal models and laboratory work which has been done to try to establish a gene therapy trial. This is what I'm going to talk about, just give you a short summary on that, and I think Dr. Smith is going to give you a detailed description what has already been achieved in the recent years in ophthalmology and in gene therapy. Now, gene therapy, when I want to describe it, it involves the delivery of therapeutic genes into the human body, into the targeted cells we want to treat, in order that we correct the disease, so that we correct these conditions which are caused by a non-functional or defect gene. This can be done by using a kind of a vector which you can imagine as a taxi driver, which takes the healthy DNA segment into the specific cells we want to treat, and these therapeutic genes are given into the target cells, and they overtake the function so that they can repair the function of these cells. Now, most of these vectors are modified viruses, which are non-pathogenic. I think this is also very important to tell, that this is non-pathogenic. They only have the ability that they can go to a specific address and deliver something, but they do not cause any further diseases, as you would expect from a virus. There are two primary strategies, ex vivo and in vivo, and of course regarding ophthalmology and regarding the eye, we will do in vivo therapy, so we will treat the cells which are in the retina. Now, why is the retina such an ideal target? It is a small and closed organ because of the blood-retina barrier, which enables and limits that the vectors can go everywhere in our body. This is a very important fact, because this enables us that we treat only one specific cell layer or one specific target cell without having the fear that it might get somewhere else where we don't want them to. Also, a very important thing, that these cells, the photoreceptors, which we would like to treat, are not dividing anymore. So we don't have to be afraid that there will be diseases which occur later because the cell is dividing and it might not taking over the information properly. And this is also an advantage because theoretically we only have to treat these cells once, and they get the information and then they can work with it. Also, a huge advantage for ophthalmology, for us clinicians, that the retina can be seen. So I can take a look at the eye, I can see the cells actually, which I want to treat here. And I can also measure the function of these cells. So I also have an information of a feedback, kind of what I have already achieved with my therapy. Just a very schematic way how to present gene therapy. We have here a modified DNA which is built into a vector. This is the part, the healthy gene we want to deliver. We put this into this vector, which is working as a taxi driver and takes our healthy DNA segments to the cells where we want them to. They also enable that these DNA segments get into the cells and they deliver it properly so that it can be used by our cells. And if we have the new coding and the coding, the information for the healthy proteins, it is our hypothesis, and it has been already shown in some clinical studies, that these proteins are then produced by the cells, which can restore the function. Now, if you want to imagine it, how it would look in a human eye, how it would look in your case, of course, it is a surgical procedure, but it is a surgical procedure which is not very long, although you have to be very careful and you also have to be very prepared. This can be done in local or full anesthesia, although this is not a very long operation, so please don't imagine that it lasts hours. What we do first is that we do a vitrectomy, which means that we take out from the eyeball, which is filling the eyeball, the so-called vitreous. This is a jelly-like structure which fills the eye. And then, if we take out this structure, then we can get to the retina. We have to prepare the retina by an injection on the place where we want to give our vectors, our therapeutic agents, and then we inject them underneath the retina. Of course, after surgery and before surgery, we do need a lot of examinations, and we do need, after the surgery, eye drops, medicaments, control examinations, just to make sure that everything worked properly. This is a surgical procedure which is not new, which has been used very often in ophthalmology, and also this specific regarding gene therapies, which is a surgical procedure which has been established and used since 2007. Now, in our special case, what we would like to do is to establish a gene therapy for patients suffering from achromatopsia who have a specific mutation in CNGA3. As you can imagine, gene therapy can only be done in those cases where we exactly know what the problem is, where the mutation is, because otherwise we cannot fix it. So in these cases, what we are trying to establish as a gene therapy trial is regarding to CNGA3 cases. This is a joint Tubingen-Munich project, so we do need a lot of expertise to work in this field and to prepare everything so that we can do a human trial. It of course needs a lot of preparation, a lot of laboratory work, and a lot of results which we have to have before we can dare to go on a human trial. Therefore, we need a lot of preliminary work which has already been done. CNGA3 has some historical reasons in Tubingen why we have chosen this gene. First of all, because it was the very first gene described for achromatopsia, and ever since we also had the first mouse model for the CNGA3 mutation. And we've been trying to establish in mouse models a gene therapy, a gene replacement therapy, which has already been reported by our colleagues and in some other labs. And we do see some very positive results which give us the hope that we can move on in a human trial and that we can restore or improve the function of the cones, which are still there in the retina but just not working. Now, what we have seen in our mouse models, we treat one eye of the CNGA3 knockout mice.
So if you take mice who are suffering from this disease because the CNGA3 gene is not working, then treat one eye. We can do, of course, some experiments and some measurements. In red, I don't know if you will see it properly, but the red curves are supposed to mean the treated eye, the black ones the untreated eye, and the gray columns just show a wild type, so a normal mouse, how the normal mice response would look like. If we do electrophysiological measurements, so objective measurements, how the cells are functioning, we see that the rods are functioning normally in every case because we do not have a problem in these rod photoreceptors. So all of them really show nice responses under dark conditions where the rod's supposed to work fine. Now, if we take a look at daylight conditions where we are measuring the cone function, you can see how a normal eye would respond to the light. This is this curve. And we can see a difference in the non-treated and in the treated eye within the same mouse. You can also see that the treated eye has bigger responses where we have a mixed response from cones and rods, and if we do see only the rod function, we also see that there are some restored function. Now this is only one thing that we can measure something, but the other question is, what is experienced, how the vision is changed? And therefore we prepared also a behavioral test for our mice, so they had to be trained first in a special so-called swimming pool where they can, where they had to swim. We trained these mice for 1 day, and we gave them a platform which was stable and marked with a red flag. Now the mice had to learn on this first day where they have to swim to survive, so that where the platform was stable. They have also connected it that this red color or this flag is supposed to mean that this is a stable platform where they can go to. On the next day, we also tested the mice, but we gave them two possibilities. A platform which was stable, which they have already known and which was marked with a red flag, and we gave them another platform which was unstable and marked with a green flag. And we did see a difference in these cases. The mice with non-treated eye had kind of a 50-50 possibility what to choose. On the other side, the treated eyes of the treated mice could really decide where they have to go because probably there was some restored vision. Now, if you want to see how a normal mouse would swim, we would see almost similar results as in our treated eye, which is a behavioral test that shows us that there might be some restored vision and gives us hope that the vision can be changed, not only the ERG responses we can measure. There are also a lot of things we have to do preliminary before getting into the human therapy trial, and at the very moment parallel to our work, there are a lot of colleagues working on it that we prove that this concept is working and that we have a significant hope for that the therapy will work and will restore the cone function. Now, what we are planning in the future is that we treat CNGA3 patients. In the very first trial, we will choose 9 patients, and only one eye will be treated with 1 injection. Of course, this will mean that we will need a lot of examinations before that. You come then to the surgery, you have to stay there for a couple of days, and then we will also have a couple of controlled examinations, so it's really also time consuming, but we do have to make sure that this is something which we can hope and which we have to work on further. In our project, the timeline looks like this. The project started in 2012. Of course, there was some preliminary work before, actually a lot, so that we could move on with this step. And now we are at the phase where we are recruiting patients with CNGA3 mutations. And parallel to us, the vectors are getting prepared, they are getting tested, because we also have to know that there are no toxic effects which we have to be afraid of. The first clinical intervention is planned for the beginning of 2015. So at the very moment, I am recruiting the patients just to give them the information that there is a possibility in the near future, hopefully, that achromatopsia can be treated. And we also do clinical examinations just to see for us how the CNGA3 achromatopsia really looks like. And on the other hand, it's also important for you patients how your retina works, like how the retina looks like, so that we have the actual data and we are up to date regarding the clinical characteristics. So in this place, I would like to thank you for listening to me, and I hope that I can welcome in Tubingen as many patients who want to perform these clinical examinations. And of course, I'm also thankful for the patients who have already visited us. And please just contact us whether it's the clinical study which you're interested in and you already know how the genetic results look in your specific case, or if you haven't had a genetic analysis of blood samples, please contact my colleague Susan Nicole. We are happy to help you with that. Thank you.